Thalidomide is a drug used in the 1950s with devastating teratogenic effects. While the precise mechanism is debated, studies have indicated that upregulation of the BMP-DICOP1 Wnt signaling pathway plays a key role in thalidomide's teratogenesis. In recent years, thalidomide has been adopted to treat autoimmune and inflammatory diseases such as rheumatoid arthritis, making it important to understand the drug's mechanism in cartilage tissue. Basal-defined chondrogenic media lacks 15 vitamins and minerals. A complete supplemented media has been proposed to mimic in vivo conditions. In this study, we explore if supplemented media alters thalidomide's effect on the BMP-DICOP1 Wnt signaling pathway in primary human articular chondrocytes and mesenchymal stem cells. Chondrocytes and MSCs were seeded in basal or complete media with or without thalidomide. Pellet contraction was studied for up to 18 hours. Huxt-stained cells were imaged every 13 minutes, while unstained pellets were lysed for RNA extraction at 18 hours post-seeding for HACs and 3 and 18 hours for MSCs. Relative gene expression of DICOP1, BMP4, axin-2, beta-catenin-1, and SOX9 was determined with QRT-PCR. In MSCs, peak pellet size difference occurred 96 minutes post-seeding. Thalidomide had opposing effects on contraction in the two medias, increasing speed during early pellet formation in complete media while decreasing it in basal. The largest difference in pellet size for HACs also occurred at 96 minutes, with thalidomide decreasing contraction speed in basal media. Thalidomide had no effect on contraction in complete media, nor were there any differences in pellet contraction between basal media and complete. For both MSCs and HACs, donors had varying levels of response to the addition of thalidomide. This difference in response may be indicative of donor susceptibility to thalidomide, as some individuals exposed to the drug during embryogenesis did not develop limb defects. Still, by 18 hours, there was a significant decrease in SOX9 expression for MSCs exposed to thalidomide, which was observed in complete media, but not in basal. Additionally, there was a non-significant but directional response to thalidomide for DICOP1 in complete media for MSCs at 3 hours and beta-catenin for HACs in both basal and complete media. While complete media did not convincingly elucidate any effects of thalidomide on the BMP-DICOP1 Wnt pathway in MSCs or HACs, it did reveal a decrease in SOX9 expression in MSCs and had a slight effect on early pellet contraction. Given the donor-specific response to thalidomide, future study may include evaluating responder and non-responder donors, assessing further time points including at the point of maximal pellet size difference, or increasing thalidomide dosage.